This is episode 61 of the Immunology Podcast, Pancreatic Cancer with Dr. Vinod Balachandran. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Rad. Welcome back to the Immunology Podcast, where we have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Immunology Podcast, rate us and leave us a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today we have Dr. Vinod Balachandran from Memorial Sloan Kettering on the podcast to talk about his research on rare subgroups of pancreatic cancer patients with exceptional long-term survival. We've also got our usual roundup of recent highlights of immunology news coming up, but first...
Jason, don't forget, IUIS 2023 is coming up this year, November, December. We are looking forward to attending the International Congress of Immunology in Cape Town, South Africa, from November 27 to December 2nd. Interested? Learn more at iuis2023.org.
Oh, I'm excited. I'll be taking the longest flight of my life to get there.
Well, good for you. So how many hours?
Well, the first one's just an hour and a half to Atlanta, but Atlanta to Cape Town is 16 hours in the air.
Oh, 16 hours actually quite, you know what? It's actually a long flight. I'm not gonna.
I actually think it's longer than your flight from Amsterdam to DC.
Oh, yeah. An Amsterdam DC flight is not that bad. It's like 8-9 hours. But, I've taken the 14-hour flight to Buenos Aires from Amsterdam. So, quite decent. I have to say that's 16 hours. But I hear that you're going to be comfortable. So that's I don't feel bad.
I am going to go in bougie class.
What a bourgeois you are.
Look, I'm old. My hip will act up if I don't have a comfortable seat.
That's true. Well, I know one of the papers today, I might have a solution for you, but let's not get ahead of ourselves.
Oh, you're going to solve my budding hip arthritis after age 50?
Maybe. At least the cognitive issues, maybe. We'll see.
Oh, the fair.
But yeah, so very exciting. And so what's new? What's coming up in your side of the world?
It's back to the regular grindstone for us. So, back to school in a few weeks after American Labor Day in early September. So the kids go back to school, which means they don't have to have different camp schedules all week for the entire summer and have Uber drivers for parents running around.
Finally, you'll get to rest a little bit.
Yeah, school is actually the less chaotic time. Also, oddly, at work with vacations, everyone takes all these different vacations that's never synced, so you can't ever get anything done for about 3 months or 2 months.
Yeah.
Like July and August, it's just like maybe the person's here, maybe they're not.
Well, I wish you the best coming back to normalcy. I still have a short holiday plan, so I will make my way down south and up to Switzerland to finally see the mountains again and enjoy thinner air for once in a while.
You're not going to go in the winter and ski?
Well, the winter is far away. I don't think I'll make it to the winter if I don't take some days off now. So I figured I'll check it out in the summer first. And then we'll see what I do in the winter. I also the mountains are great for summer. I don't know, there's not only a winter destination.
Oh, they're nice. True. But my goal is to ski the Swiss Alps in the winter. It's one of the things I want to do. And I haven't yet.
Well, just text me. We can go in January, February. Just let me know. I'll do some scouting during my time there. We will be staying at a ski slope in a ski resort that is not for skiing right now. But yeah, it's just mostly about being above sea level for a change. And I was hoping for better weather, but doesn't seem like it's gonna be the case. But it doesn't matter. In any case, why don't we get started?
All right. So how are you going to fix my memory, Brenda?
I might have found the fountain of youth. Well, I'll get started today because I think you're very curious. And this story, it's always important, very cool. There's certain types of stories that sometimes you need to always keep a skeptic eye on because this story is pretty interesting. And, I think a lot of people are behind results like this. So basically, the paper I am talking today about is called Platelet Factors Attenuate Inflammation and Rescue Cognition in Aging. So First author Adam Schroeder and Patrick Ventura from the lab of Saul Villera and at UCSF at the University of California in San Francisco. It's published in Nature on the 16th of August. And you know, I I want to tell this story, but always I think it's important to take it with a grain of salt. There is obviously a patent associated with this research, and there's a lot of billionaires around, looking for the fountain of youth. But it's interesting, and I think we'll see how the story evolves with time. Basically, in the story, they are studying a well-known phenomena by now, which is the fact that if you share um, blood between a young and a young individual with an old individual, the old individual benefits from the quote unquote young blood. And this has been shown in mouse systems with blood transfusions, but more specifically, it seems to be plasma. The plasma components are sufficient to mediate most of these kind of rejuvenating effect of young blood. And there has been some, models suggesting, that it can reverse impairments like age-related impairments in neurogenesis, synaptic plasticity, cognitive function, all the whole suite of things you want to get your brain better when you age. So that's what I mean. I have to say, disclaimer, I'm a little bit skeptical. I think there's a lot of interest, a lot of if you read Twitter, like half of it is about billionaires trying to make themselves younger. My epigenetic age is 8 years old or something like that, which I think is a little bit absurd. But let's go to the facts. They have these mice. Basically, they have old mice, young mice, and they prepare from the blood of young mice. Like vampires, they make blood plasma preparates, and they have a version with and a version without platelets. And they have as a control kind of saline infusions. And they inject old mice over 24 days. So it's a fairly short intervention, I would say. And they we focus on the hippocampus, which is region of the brain that has been apparently suggested to be particularly sensitive to aging, and it has to do with memory and many of the things that are first start to decline as we age. And what they see is that they do all this RNA-seq, and they see particular genes, and especially some genes related to an inflammatory response that are differentially expressed compared to saline injections. And they see a decrease in some of the genes they mentioned, and that kind of pop up throughout the work are TNF, a gene from a complement gene called C1QB and CD11B, this is myeloid marker. And they see other inflammatory markers, or they have a list. They do this whole analysis. And they kind of use this as a model. So here's what we see. We see reduction in inflammation in the hippocampus in these all aged mice when we transfuse them. When you inject them, not even transfuse, they're just injecting plasma from young mice. And so they zero down into one particular molecule, which is a chemokine called PF4, which is released from platelets and is involved in coagulation and has immunology function. The truth is that they kind of jump into PF4. It's not really clear why they jump into PF4. They just it is known that PF4 is part of, mediates part of this inflammatory response, and they just zero in on it, and they really continue their work on PF4. So again, I feel like they're just trying to justify working with PF4. That's probably where the patent is. And they say that it has been involved in the beneficial effects, for example, of exercise, that it is known that it has, anti-aging properties, things like that. And they show that old mice have less PF4 than young mice in their plasma, and they verify it in their own experiments. So basically, they start administering the mice with PF4. And it looks like this is decreasing some of these inflammatory markers that they see in the hippocampus of the aged mice alone, without needing to do the whole plasma thing. And they look at several things, and basically, they try, they see certain genes that are inflammatory genes that are downregulated, and they seem to have some improvements in synaptic plasticity, probably. And when it comes to the immune system itself, they see that some of the markers that are some of the changes that are associated with an aged immune system seem to be at least partially reversed by the injection of this PF4. Particularly the increase in the myeloid lymphoid ratio. So usually you lose lymphocytes as you age, especially also naive type of lymphocytes. You do end up accumulating kind of effector memory lymphocytes as you age, as characteristic of an aged immune system. And it's amazing. There's a lot of these markers seem to be reduced. I mean, not gone, but they do seem to find differences pointing towards a rejuvenated, quote unquote, immune system when injecting PF4. They also had a PF4 knockout mice, and they see that, for some of these markers that they see increase in age, that it is worse that PF4 knockout mice have like higher levels of some of these markers, including C1q and CD68, microglia with CD68. So, when it comes to like the I think one of the major issues, which is kind of the cognitive benefit of this treatment, I guess it's hard to make cognitive analysis on mice. It's not like you can ask them, what did you eat yesterday? Do you remember the string of numbers? But there are some more or less standardized methods of analyzing cognitive function in mice, and they seem to find some levels and some of their analyses, which I'm not an expert in mouse behavioral studies, but they do seem to find some, I think, modest differences. But I'm not so familiar with how these experiments usually go. And when it comes to how the mice react to their environment, they see one particular thing that they do is this novel object bias, in which the mice are very attentive to our new stuff, so they notice things that are new and they give more attention. This is a kind of a marker of cognitive function, and they also have some memory ways of measuring memory. And they see some, I think it's modest, but they see some improvement in this cognitive function studies that they do in the mice. So basically, they really show that they make the point that this PF4 is a major mediator in the progressive loss of PF4. Is a mediator in this inflammation that affects the brain of older mice, and that it can be at least partially reverted by adding PF4 on top, like just injecting PF4. And they show, using CXCR3 knockout mice, that some of these effects of PF4 is mediated by CXCR3 that is expressed on immune cells, and that's probably partly how it's acting on the immune system. So basically, that's kind of the idea. So I don't know, maybe we're gonna start all injecting ourselves with PF4, to prevent cognitive decline. What do you think? Would you take it?
It's already happening among those billionaires.
Yeah, I mean, there's this whole story of like Jeff Bezos, buys plasma from young people and injects itself. I mean, I don't think that's true. I don't know at this point. I hear such insane things sometimes.
I think there's definitely people who inject plasma, or especially to damage joints and stuff, to try to get the cartilage to regrow. That is
really okay. Yep. Well, this is the next thing. They're gonna just be injecting PF4.
Now you can go recombinant. Oh yeah.
Yeah.
You get your home PF4 kit to purify your own recombinant protein.
Yeah. Just make sure you don't have any LPS contamination on that injection, because that's gonna suck if you put it in your cartilage or something.
I don't have a segue here because I'm talking about influenza.
I mean, it's bad for old people. It's also,
maybe, yeah, it has to, like, I guess I need my flu shot because I'm aging too. Sure, we'll just keep piling on me. All right, so this is in Nature Immunology. It is published August 17th. Its baseline innate and T cell populations are correlates of protection against symptomatic influenza virus infection independent of serology. First author is Robert Medelman. Last authors are multiple Richard J. Webby, Q. Su Huang, and Paul G. Thomas, and the Shivers 2 investigation team, because someone has an acronym for some study here. trying to parse out this paper, it's both interesting and, coming up in Nature Immunology, and also it's one of those papers I'm surprised we didn't already know this about. And so it's a little bit duh, but apparently no one's ever done the work to determine this before. So they took 206 people that were either vaccinated or not vaccinated for influenza in different cohorts and looked at their immune cells over time, pre-infection, post-infection of influenza, symptomatic, non-symptomatic. So that's where the real cool part of this is. This data set is really awesome, right? You have a really cool data set, which I think is most of it. So if you like this or need this type of data for your research, you should just go get the paper or pull the data for it. Because the conclusions I'm going to get through real quick, which is that vaccine-driven immunity is largely highly humoral, right? Very B-cell driven. But the number one protective thing they see for preventing symptoms, while B cells help, the number one thing we see for preventing any illness at all is actually adaptive T cell immunity and some innate immunity, but the big signals are from some CD8 cells and some CD4 cells, depending on the subsets. The biggest theme that kind of popped up was CD4 and CD8 T cells. So cellular immunity versus humoral immunity, right? So humoral immunity, definitely, we know influenza vaccines and just humoral immunity in general will prevent you from getting as sick, but it doesn't seem to prevent you from getting sick. But if you have T cells already primed against the flu of choice, either through a previous vaccine, but most of the vaccines aren't very good at it, or you've had it before and you just are that way now, your outcomes in terms of do you get sick or not are much, better. And so the real point of this is that maybe vaccines for flu should, instead of being just this humoral strategy, should also start looking at ways to generate some cellular immunity as well, which is arguments for things like mRNA-based T-cell vaccine or mRNA-based influenza vaccines and other such. So that's real take home from this, is that cellular immunity matters too, and that our vaccine strategies up until recently have largely been humoral in nature, and that has some inherent drawbacks for some of these respiratory viruses. You don't get the full shebang from it. So there you go. That's distilling it down. But if you want this data set of every cell population from different patients tracked based on did they vaccinate or not and status of symptoms or not, they have that in this paper. And that's what the paper's bulk of the data is, but not the bulk of the conclusions.
Yeah, I mean, I guess after all I've read about the response to SARS-CoV-2 and how much T cell immunity has been kind of propelled to the attention of people lately, I'm not surprised. But
right. But interestingly, not done yet in flu until now. So there you go.
Okay. So this flu vaccines, what is the format that these vaccines have that they study?
That's the standard IM flu vaccine.
Intramuscular, they're but sorry, are they recombinant or are they inactivated virus? What are they?
Oh, I think flu is dead virus, if I remember right, that's grown up in eggs.
Oh, yes, of course. Yeah. Okay. So really attenuated.
Not attenuated, dead.
Okay.
They can't come back. No attenuated coming back resurrection. It's just dead.
Dead virus. I mean, can a virus be dead if it was never really alive?
We should have a whole podcast where we discuss the living nature or not of viruses.
Oh, no. I think there are other podcasts that do that regularly.
But we haven't done it, Brenda.
No, that's true. We should get a virologist just to have fun with it.
You should. That'd be a great episode.
All right. Then let me talk about my second story of the day. Also very interesting, a little bit quite complex, and also I think a great resource for people studying this. This article is called "Mapping the T Cell Repertoire to a Complex Gut Bacterial Community," so it's down your alley, I think.
Right up the alley. Oh yeah, this made the rounds in my workplace.
Sorry, listeners, up your alleys, not down your alley. Oh gosh, this is English prepositions. Okay, so um, first author Kazuki Nagashima from the lab of Michael Fiesbach from the Stanford University. And I mean, again, this is another story about, one of my favorite subjects. How do T cells see the world? And in this particular case, is how do T cells interact with bacterial colonization in our guts? And we know, of course, that bacteria in the gut are very important and they're part of the normal development of an immune system. You need bacterial colonization for a proper development of your adaptive immune response. And we do know that, T cells must be recognizing something on this bacteria. And we also know that certain bacterial strains are more likely to elicit T cell responses. And oftentimes many of the responses are, symbiotic and are good. And you develop a healthy repertoire of T cells in your gut. This is part of your normal development. And so, but what is not really easy to find out is exactly how are the T cells seeing the bacteria, especially given the fact that bacteria are not eukaryotic cells. They are not presenting antigen. But we know that there's many, mostly CD4 helper cells that are responding to their presence. So clearly there must be peptides being presented on, antigen presenting cells, and they're being recognized by specific T cell clones, and that they are responding to this signal, and they are establishing themselves in the immune system around the guts and the colon and the small intestines. So what they do is like, how can we understand exactly what these T cells are seeing? How can we kind of pick apart a little bit the complexity? Because of one of the big issues with microbiome studies, and as you well know, is that normal microbiomes have huge complexity, and often this complexity is very important to how the whole thing works out. So even there's not the saying to study a mono, like just a mouse colonized by one bacteria is going to give you completely different results to the normal situation, which you have, thousands of different types of bacteria colonizing your gut. So what they do is that they generate they colonize germ-free mice with specific bacterial communities. They start with 2 different ones that have roughly 100 different bacterial strains, defined, well known, and they use this to study the T cell repertoire that occurs after the establishment of these bacterial communities. And basically, they quantify that they look at the T cells in these mice and the helper T cells and the regulatory T cells, and they're trying to understand how many of these are responding to specific bacterial antigens, how many of these and what kind of profiles, T cell profiles, you obtain as a consequence of it. So there's a lot. They do a lot of analysis and a lot of work. And I'm not going to go a lot into details because, I mean, it can get a little bit dense. But what I thought was really cool is that what they see is that, in fact, when they can find, within the expanded T cells, they find T cells that are they sequence many T cell individual T cells, and they find certain clones that are expanded. Not a lot. Some of them is like 2. For it to be expanded, it has to be present at least twice. So it's not a huge expansion. But they do see that from all of these clones that they pick up, they can show that some of them do recognize bacterial products. So if they prepare hybridomas from these clones and they co-culture them with products from all the different bacterial strains presented on IPCs, they show that at least some of these clones are, demonstrably recognizing bacterial peptides. And I think what I thought was very cool is one of the families that affect that have, so there's clearly some, they are not species specific, this recognition, So you have one T cell clone that is going to be recognizing products derived from more than one bacterial strain, which shows that they're recognizing some shared antigen. And I think for me, the most interesting part was this Firmicutes derived antigen. And it was very widely expressed, and it was very widely recognized by T cells. And we know that this particular genre, the Firmicutes, is very well known to interact with the immune system and to colonize the gut and to really initiate the proper maturing of the immune system in the gut. So basically what they see is that what is the antigen that has been recognized, and they can go all the way to pinpoint one specific protein, one epitope that is derived from a protein that is very highly expressed in these bacterial proteins, and that is very antigenic. And most of the T cells that are recognizing these bacterial strains are particularly picking up this one peptide from this highly expressed protein in this bacteria. And this is kind of shared by the whole family, by many bacterial strains. And that this is a major immune recognition site for T cell responses after colonization by this particular bacterial strain. But and I think although it seems like a rather modest result, I think it's really cool because it really gives you a kind of straight line between the kind of antigens being expressed by the bacteria and how this affect the development of the T cells in the area. And they also see that for certain bacteria, for certain antigens, they can see that some of the T cells, they don't always, the way they develop, some of them become regulatory T cells, and certain antigens seem to have a tendency of inducing more regulatory T cells than others. And this is probably part of, like, in a simplified way, what happens when you're colonized by, just wild type regular bacterial communities. And I think it's really cool because it tries to reduce a little bit the very large complexity in a normal setting, but still gets really clear results. And I think it's a great way of understanding how the gut microbiome affects adaptive responses and the kind of the homeostatic condition in the gut.
Well, all I'm going to say is, because this ventures into work territory to me, is I'm a very big fan of Firmicutes. So it's cool to see how they're sculpting the immune system.
Yeah. Yeah.
So this was pretty seminal work in the microbiome field that popped up. So I am honored that you covered it for me.
Well, it was a pleasure to read.
And it had T cells. So it's really the combination of both worlds.
Right. I like it because it's like, yeah, there's microbiome, affects the immune system. But this is a really clear mechanistic way. Like, you really shows you what happens under the hood in a way that you can understand and you can quantify and you can, trace back. I really like that. It was a very elegant set of experiments. I really liked it.
Yeah, that's super cool. All right. So last paper of the day, also in Nature, endothelial aryl hydrocarbon receptor activity prevents lung barrier disruption in viral infection. First author is Jack Major. Last author is Andreas Wack. Also coming out here on 16th of August. So if you're not in the know, the aryl hydrocarbon receptor, AHR, this is a receptor that's often activated by dietary supplements. It's had or just things that you eat in your diet, indoles. And it's had a lot of research in it lately. There's a microbiome link with it. There is a component where it's really important for barrier function in the gut and quieting down the immune system. And so they're taking this notion that, hey, it's important in the intestine. What about other epithelial tissues? And there's a lot of crosstalk. There's a lot of similarity here. And they do a series of elegant experiments, both knocking out the enzyme that turns off the molecules that signal this receptor, which is a cytochrome. So getting rid of the metabolism of the ligand so that the ligand's more active, and then using the knockouts of the receptor and then tissue-specific knockouts and inducibles, pick all that, to figure out what this receptor is doing in the lung. And they identify that it's actually most active not in epithelial cells in the lung, but in the endothelial cells, those vessels that transport blood back and forth. And there's a lot of them in the lung, obviously, because you're having air exchange with your blood. So there's lots of endothelial cells. And they isolated the fact that during influenza infection, influenza in general dampens the aryl hydrocarbon receptor response, leading to lung wall leak, inflammation, and all of the badness you think of with interferon 1 signaling. If you tune up the aryl hydrocarbon receptor signaling by knocking out the cytochrome that metabolizes it, you get protection from injury in the lung, from influenza. And if you knock out the receptor, you get worse injury in the lung. If you feed mice a diet high in the metabolites that you're in the pro molecules that generate the signaling when metabolized in your gut, you get more protection. And if you knock out the receptor, you lose that effect. And then they isolate this to some transcriptional networks that's called an apelin peptide system as being one of the downstream targets of the aryl hydrocarbon receptor that's really important for this process in terms of maintaining gut and maintaining lung and epithelial integrity and dampening lung tissue inflammation. So essentially they're finding that this is a central molecule, but that it's activated on the endothelial cells, which then signal to the epithelial cells of the lung to hang on tight and not leak and inflame and all of that. And there you go. So trying to sum it up, it's worth read. It gets in, they have some beautiful IF staining. They really map it out using their mouse models. It's actually only a 4-figure paper, so it's a shorter Nature paper, but it's to the point and really gets into how it works.
Okay. So these mouse models, are they kind of like complete knockout or like specific knockout in the...
They've done both. So they showed that if you do it in both, or if you knock it out but in the endothelial cells, it's the same effect. To the point being that whatever else is in the rest of the tissue does not in the mouse matter here.
Right.
They did knockouts. They did inducible knockouts so they could turn it off after infection and watch the mice get worse.
Mm-hmm. Yeah, because I guess it's also very important, especially in the case. So it's something that's intrinsic to the endothelium and not mediated by immune cells that lack AHR. It's just literally those cells that are...
Those cells are signaling through it, creating downstream signaling that affects the environment around them.
Okay, got it. All right. So, and what are we all getting? Do we need to get some agonists?
They're looking at it. It's becoming quite the drug target lately. It's kind of the next frontier of immune regulatory drug targets is AHR, at least in my opinion. It's being talked about quite a bit.
Yeah, I mean, and also I think this idea of reducing inflammation, especially in the lungs, like even if it's an infection, like in SARS-CoV, I think we've learned a lot that how important it is to reduce inflammation to keep the tissue, because even if the inflammation is too much, inflammation is maladaptive. I mean, it's part of the immune response, but too much of it is not good. So you need to get that checked.
Indeed. No, I completely agree. It's all about regulation. Yeah, I don't have a fun segue here either. We're segueless today. I guess the company also went out of business, so maybe we can.
The company went out of business.
Yeah, I don't think they make those Segway bikes anymore.
Oh no, it's not. That's not the company. I think you're talking about the electric bike company that went out
No,
of...
the ones that you lean forward on. They're not bikes, but they're those like there's those platforms that look like a T with the 2 wheels.
Yeah, that
lean.
Segway, they did go out of business. Yeah. Who would have thought those 2-wheeled transport, whatever they were not as
I know
long-term
success. Because I really
success.
wanted to Segway joust and I never got to.
Segway joust, yes, yeah, I'm sure you did.
You can imagine what that would look like.
Yes, ridiculous. That's how. Why don't you adjust in bikes? You can get some bike adjusting, and then
Oh, that sounds so much worse. Anyway,
I mean, can't you bike without your hands? And
you
it's the
can for
same.
short periods of time, I suppose. All right. Well, we're going to be speaking with Dr. Vinod Balachandran at Memorial Sloan Kettering Cancer Center in just a moment. But before we get to that, this week we'd like to remind our listeners about Pancreatic Cell News, one of STEMCELL's free weekly scientific newsletters. Pancreatic Cell News summarizes all the latest research, news, jobs, and events in pancreatic cell research and delivers it right to your inbox every Tuesday. Save time and keep current with Pancreatic Cell News. Subscribe for free at pancreaticcellnews.com.
Hi, everyone. Welcome to our second part of the show, our interview. Today, we are talking to Dr. Vinod Balachandran, who is an attending surgeon and the lab head at the Human Oncology and Pathogenesis program at Memorial Sloan Kettering Cancer Center in New York City. His lab studies immunotherapy of cancer, and I personally really like his recent work on a clinical trial using mRNA vaccination to treat pancreatic cancer, which is I think a very critical cancer type with very little treatment options. So I'm very excited to talk about this trial, how to target neoantigens of pancreatic cancer, and what he has learned from his work on this topic. Dr. Balachandran, thank you so much for joining us today.
Thank you for having me.
Thank you for being here. All right, Brenda, you're really excited, so please take the first question here.
Well, I guess it would be nice to get our listeners settled on a little bit of what your research is about and what excites you about. I would start with your recent phase 1 trial, which you published the results a couple months ago, and why is it important to look into neoantigens in pancreatic cancer and maybe other types of cancer?
Sure. So as you sort of alluded to already, pancreatic cancer is one of the leading challenges, I think, in all of oncology, soon to become the second leading cause of cancer death in the United States by 2025. And this is largely because the current treatments for pancreatic cancer remain surgery, chemotherapy, and radiation. And the more recent advances in cancer therapeutics, namely targeted therapies and immunotherapies, really have not been successful at all. So because of this, we are still sort of stuck in treatments from many decades ago, and survival rates with pancreatic cancer, despite our best current treatments, remain only approximately 10%. So it really points to an urgent need for new therapies for pancreatic cancer and really urgent need to improve outcomes. So this has really been our motivation to study this disease.
So you started some years ago. You showed that neoantigens could actually play a role in how the immune system targets pancreatic cancer. You looked into patients that had responded. I don't remember if it was for immunotherapy or that actually had progressed better on upon therapy, and you took that learning into a clinical trial using mRNA vaccination with neoantigens, with personalized neoantigens. So maybe can you tell us a little bit about what did you learn about this? How do these neoantigens, maybe talk a little bit about where they come from and how do they relate to pancreatic cancer survival, and how did you think about harnessing this for therapy?
Sure. So as I mentioned, about 90% of pancreatic cancer patients die despite our best current treatments. But remarkably, there's a 10% of pancreatic cancer patients who get the same treatments as everyone else, but survive long term. And why this was unknown. There had been several hypotheses been proposed, but really more detailed immunological mechanisms of a potential immune-mediated long-term survival was really not known. So back in 2017, we published a paper on this where for that, what we had done was systematically collected tumors, blood of these patients and looked to see what might be different about them compared to the other 90% of pancreatic cancer patients. And the main take-home message from what we found in that paper was, number one, when you looked at CD8 T cells in their primary tumors, in these long-term survivors of pancreatic cancer compared to stage and treatment-matched short-term survivor controls, there's around a 10 to 12-fold increased density of CD8s. So they have a lot more CD8s. But this per se was not that surprising, and others had demonstrated similar results in the past. But the antigens were not really known. And the prevailing belief had been that lowly mutated tumors like pancreatic cancer were unlikely to generate mutation-derived antigens. And this was sort of the gestalt at the time. We thought perhaps, given the fact that tumor mutational burden does not very strongly correlate with spontaneous immune activity, which would suggest that not all mutations have equal immunogenicity, that in lowly mutated tumors, perhaps this might be the ideal spot or clinical scenario to, number one, see if mutation-derived antigens could be immunogenic in a lowly mutated cancer, and number two, develop potentially an antigen hierarchy you could then apply it to more highly mutated tumors. So what we had found was that we think, contrary to what the prevailing belief was, that the spontaneous immune reactivity of T cells in these patients' tumors were directed at mutation-derived neoantigens, which were primarily passenger mutation-derived. So I think this was the motivation to the hypothesis that if this is what is happening in the best case scenario, if you will, namely you have a spontaneous immune response to a passenger mutation and you have long-term outcome, could you replicate this with a therapy by delivering the antigens?
So talking therapy now with this phase 1, you're using a drug, as far as I can tell, this autogene, I'm not going to pronounce this right, Sivumerin. Did I get that right in pronunciation?
Close enough, yes.
That is something you guys are developing yourselves in-house at Memorial Sloan Kettering. Is that my understanding is correct?
No, this was a collaboration between Memorial Sloan Kettering, Genentech, and BioNTech. So the drug here, in this case, autogene Sivumerin, which is an individualized uridine RNA lipoplex nanoparticle vaccine. This was developed by BioNTech.
Yeah, I was going to ask how you were going to be manufacturing drugs for people as a university, which I know you can do. It's just very hard, being a drug manufacturing myself. And then you answer the question, which is you didn't, which is the much easier answer.
Right. In this case, this was a very productive partnership between MSK, our group at MSK, and the groups at Genentech and BioNTech. Yes.
I remember reading that production started around the pandemic. How did you guys manage that? Because then I guess all the mRNA production capacity was diverted to a maybe more acute issue at the moment.
It was certainly an interesting time. We published our first findings of spontaneous T-cell reactivity to mutation-derived neoantigens in long-term survivors of pancreatic cancer in 2017. And that was the motivation for this clinical trial of individualized mRNA vaccination for pancreatic cancer. And the reason for individualization was because these targets that we found to be immunogenic in these long-term survivors were passenger mutation-derived. We did look for driver mutation-derived reactivity, but we did not find any, at least as the dominant endogenous responses. So our philosophical view on this was that the vaccines the ideal vaccine antigens would mimic the endogenous antigens. So for this would require individualization of the vaccine. And we thought that the ideal platform for rapid custom cancer vaccination in the clinic was to use mRNA. So this was the genesis behind this collaboration between our group, BioNTech and Genentech. So we designed the trial. We, in fact, treated our first patient on this clinical trial in December of 2019. So this was before the pandemic. And shortly thereafter, found ourselves having to conduct this complex trial in the midst of a pandemic. And this trial was complex because, what it requires is we do surgery on pancreatic cancer patients to remove their cancers here in New York. And then within 72 hours, we ship their tumors to colleagues at BioNTech in Mainz, Germany, who then sequence the tumors, identify the neoantigens, and custom manufacture the individualized vaccines. So while that's ongoing back in New York, we treat patients with their first treatment, which is anti-PD-L1 blocking antibody. This is at 9 weeks after surgery, at 6 weeks after surgery, rather. And then at 9 weeks after surgery, they received the first of their custom-made vaccines.
9 weeks is a very decent time, I would say.
Right. I mean, one of the reasons for selecting the RNA platform was because for individualization, we felt this platform could be iteratively improved and accelerated. We suspected at the time that RNA vaccines could be made very fast. Others had shown this already. We were convinced, or we were certainly encouraged by their findings. And we now, I think, know on a more spectacular way that you can make an RNA vaccine very fast. This knowledge was not available at the time at which we designed the trial and we initiated it. But thankfully, this knowledge became available through the trial. So actually, I was mentioning that the first patient was treated in December of 2019, and then in 2020, we had the pandemic. As you know, New York was quite affected by this. This was an epicenter of the pandemic in 2020, which caused a lot of clinical disruptions for us. We had hospital shutdowns, research institution shutdowns. There were also global supply chain disruptions, which was quite important for this trial, which involved real-time cross-Atlantic transfer of patient material and drug. And of course, like you mentioned, BioNTech had the added distraction now to make vaccines to save the world from COVID. But actually, thankfully, despite the challenges by the pandemic, the teams were quite fantastic and were in fact able to not slow the trial down during the pandemic, but in fact accelerate the trial to complete it a full year ahead of schedule. So this was a testament to the team's commitment to this.
Yeah, well, I was going to add is that usually your phase 3 and then your phase 3 manufacturing, you want to look like your commercial manufacturing, and that's often done on a whole other, I don't want to say manufacturing, but a whole other universe of manufacturing than your phase 1, 2. And sometimes phase 2 is a bridge. So I'm not surprised BioNTech could deliver, for instance, in this case, right? Because you're going to have, and also Moderna is not involved in this part of it, but like fundamentally where you're making your phase 3 for their one trial, because they just did one trial, right? They did a big phase 3 and went through, essentially. I mean, they did earlier ones, but like the big thing that saved the world, that whole plant and everything is over in the corner, only doing that, and is made to scale for that, because you apply for your license with the manufacturing that you're going to use, versus like small scale, small batch.
Yeah, it's not like you're competing for the same infrastructure in particular.
You could be competing for the lipid nanoparticle reagent to get to the supply chain thing, but you're not going to compete for the people, but you're not going to compete for the space in the manufacturing line in the same way, which is an interesting thing I've only come to appreciate recently.
Before we move on from this, so what did you see? How did this trial? What are the results of the trial?
So the main questions we wanted to answer in this trial were safety, feasibility, and immunogenicity. First question, were the vaccines safe in pancreatic cancer patients? The answer to this was yes. This was no surprise, because we now know mRNA vaccines have a very good safety profile. And this study confirmed that for the individualized neoantigen vaccines, where the targets are in fact individual for every single patient, so every patient's drug is an N of 1 drug, this favorable safety profile is similar. So the drugs are very safe. The second was feasibility, namely, can you custom make an individualized vaccine fast for one of the most challenging of tumors, pancreatic cancer, which is lowly mutated and stroma rich. So these are additional barriers to potential antigen selection and manufacturing and incorporation. And our a priori thinking here was, well, if you could do it in pancreatic cancer, hopefully this is a proof of principle that this could be done in virtually any solid cancer with sufficient mutational burden to detect. And the short answer for the feasibility question was, it is feasible. Our achieved time to vaccination was within 3 days of our benchmark time, which was 9 weeks after surgery. And this is the first iteration of this experience of individualized RNA vaccination in pancreatic cancer. So I think, and also we are in the earlier stages of our learnings on this. So as we continue with additional clinical trials, I think our ability to make rapid custom vaccines will get faster. The third was immunogenicity, namely, can vaccines stimulate T cells to recognize mutation-derived neoantigens in a lowly mutated tumor, pancreatic cancer, historically thought to not contain such antigens? And the answer to that is also yes. The mRNA vaccine platform was able to stimulate T cell responses in half of the patients, so 50% of pancreatic cancer patients. It was 8 of 16 vaccinated patients in this trial. And not only were the vaccines able to stimulate T cells, the magnitude of the response was very substantial, reaching as high as 10% of all peripheral blood T cells. So we think this was quite encouraging. And furthermore, when we looked at the correlation between immunological outcome and clinical outcome at a follow-up of 18 months after surgery, we found that the 8 patients who received vaccines and had a strong immune response did not see signs of their cancers recur, in contrast to the 8 patients who received vaccines and who did not generate a strong immune response, in whom 6 of 8 of these patients had their cancers recur on average 13 months after surgery. So suggesting maybe vaccines could delay recurrence of pancreatic cancer after surgery. But this, of course, is a small phase 1 trial, does not establish causation at all. That's an interesting correlation which needs to be further investigated in a follow-up clinical trial, which is actually currently open and enrolling in MSK.
I see that as a common theme in immunotherapy trials, that if strong immune responses usually correlate with good, what's the word, with lack of progression or lack of recurrence. So of course, as you say, it's a small trial, and you haven't followed up these patients for so long, but we have already results from immunotherapies of other tumors that have started many years ago, and many of these patients that they responded quickly and then they stayed cancer free for a decade or something. So
yeah, I think you're bringing an important point to whether this in fact represents a difference in host biology or in fact an efficacy of the therapeutic.
Mm-hmm.
And this cannot be deconvoluted in the phase 1 trial. The only way to rigorously answer this is in a randomized trial, which is what we are currently doing right now. But what was interesting to note in this trial was to answer the question of could perhaps the patients who received the vaccines but did not generate an immune response, these non-responders, perhaps they were non-responders because they just had poor host immune fitness, and the differences in outcome really just is a reflection of differences in host fitness. So to answer this, we looked to see whether or not these non-responders were able to mount immune responses to a concurrently administered unrelated mRNA vaccine, which was SARS-CoV-2 vaccination, because they had also received SARS-CoV-2 mRNA vaccines. And interestingly, these non-responders, even though they did not mount an immune response to the experimental vaccine, they mounted equivalent immune responses, both cellular and humoral immune responses to the unrelated SARS-CoV-2 vaccination. So our interpretation of this was that differences in host immune fitness did not seem to correlate with the observed differences in clinical outcome. But you're bringing a very important point.
For the responders versus non-responders, do you have any sense or clues about what makes someone one way or the other in terms of like HL7 or HL typing or other genetic markers that you happen to do on people? Or is that a paper that's coming out and can't tell me yet? Or
we actually did include our initial thinking on this topic in our published paper, in that this individualized neoantigen vaccine, which was based upon systemic, so intravenous administration of uridine mRNA lipoplex nanoparticles to allow for total body-wide lymphoid organ targeting. So one feature of this systemic body-wide lymphoid targeting agent is that one of the organs that it requires for maximal immune response is the spleen. And the BioNTech group actually had published evidence of this several years ago in preclinical models that splenectomy abrogates or reduces rather the magnitude of the immune response by about half. Now, interestingly, for pancreatic cancer, for surgery, there are two types of surgery that we do. One, which involves removing the spleen. So it was, even though this is a small clinical trial, it was interesting to note that the non-responders were preferentially enriched, not statistically significantly because of the small sample size, but interestingly enriched in patients who had had their spleens removed. So based upon the data we had from this trial, our current thinking is that the reason for the 50% response is perhaps because of inclusion of patients who had splenectomies. So in the follow-up clinical trial, this clinical trial is focused on patients who have their spleens intact.
But the splenectomy did not affect the response to SARS vaccination.
That's correct, because this is an intramuscular vaccine.
Right.
So there, the draining lymph node basin is unaffected.
That's a very interesting example of how the route of administration has such a strong effect on how the immune response is mounted.
Right. Right.
A human model.
Right. It was not a complete correlation. So there were patients who had had their spleens removed who were still able to mount a response. But as I had mentioned, there was preferential enrichment with splenectomized patients in the non-responder group. So, but I think we would really need larger studies to more accurately characterize this.
So you have another trial going on. So we can't talk a lot about it other than there's a trial going on right now. But part of this work that I think is really interesting is that you come at it as a surgeon, and surgeon as a career and having a lab as a career are not as common as other pathways in medicine. So I was wondering if you talk a little bit about how you ended up there and also how you manage that, given the immense clinical burden that surgery involves. And especially now you're doing clinical trials on drugs, which is very internal medicine-y.
Sure. On that topic, I've really been fortunate to have had the privilege to have phenomenal mentors who have been surgeons and scientists who have really sparked the scientific curiosity and drive, as well as shown me the power of what a physician scientist and a surgeon scientist can do. So when I did my postdoctoral training here at Sloan Kettering, I did a lab run by a surgeon scientist, Ron DiMatteo, who studied dendritic cells here in the immunology program. And he was the first person who was really able to show me the power of lab science and the ability to translate lab science to the clinic. At the time, I also had the privilege of being here when immunotherapy was in its early stages of clinical application of the first successful immunotherapies, but this was prior to them being successful. So this was during the times of the clinical trials of the first clinical trials of ipilimumab, CTLA-4 blockade, done here at Sloan by Jed Wolchuk, another physician scientist, when Jim Allison was here at MSK. And I had really the fortune of being a fly in the room when they were showing some of these pictures of patients receiving single doses of ipilimumab and having metastatic melanoma completely regress just after single doses in weeks. And I think these experiences really taught me how a physician scientist can have a tremendous impact on patients. So I think I've really been fortunate to have mentors throughout my career who have been surgeon scientists. Subsequent to those, several others have really contributed to my interest and my path. Stephen Leach was another surgeon scientist here at Sloan Kettering, who is a former director of the David Rubenstein Center for Pancreatic Cancer Research, who also influenced me in my path. So I think short answer is find good mentors.
Sound advice, for sure. So it has been a really interesting conversation. I always so excited to hear about the immunotherapies of all types. So I think immunotherapy of cancer is having an amazing time at the moment. There's so much going on and so many clinical trials of so many approaches to immunotherapy. And I think there's other types of immunotherapy for other types of diseases will also come. But I think we are reaching the end of our conversation. And we'd like to ask our guests to tell us a little bit about themselves. And so there's a very popular question that we pop up, which is, if you were not a very successful researcher surgeon, what would you be?
Well, my initial desire as a child was to be a veterinarian. I loved animals, and that was sort of my first passion. And I think later on in high school, in college, my interest morphed into more quantitative sciences, and I became very interested in math and physics. And I actually did my undergraduate degree in physics. And I was very close to pursuing graduate studies in physics as a career. So I think if I did not go into medicine, then that would have probably been my path of graduate studies in physics.
I was to make some joke, the whole Oppenheimer movie, but I don't know. I don't know. We're gonna build some atomic bomb, but that sounded more funny in my head.
Yeah, hopefully my contribution, if I had taken that path, would be slightly different. But
yeah, I was going to ask, well, what part of physics in particular really were you going to do?
Well, the aspects of math and physics that I found very fulfilling was the ability to solve a problem really accurately. So you can write an equation and solve it, and you have an answer, and you can check it, and you know it's correct. I found that very satisfying, and that was sort of, I think, one of the core reasons that interested me in that field.
Then why are you in biology? That makes no sense.
Well, I'm hoping that we can try to have similar. Biology has its own challenges. It's so much more complex, and it's oftentimes you don't get that a simplistic answer that you can get in quantitative sciences. But I think that's part of the challenge, to perhaps frame the question in a way that maybe you can get an answer that is more clear, and then gives you a path forward. So I think this is one of the reasons why I find biology challenging and thrilling.
I think that takes care of it then. And I also agree. I like the cleanness of physics and math, but like the challenge of the fact that biology isn't that way yet.
Right.
So if you're looking for postdocs, you can advertise them here before we go, or if you're looking for positions, you can put a shout out. We're
always interested and excited to recruit new people to our team. We love curiosity-driven science. We love team science. We love to work together. And our group is very passionate about what we do. So certainly, if you're interested, please shoot me an email. We'd be happy to chat more.
Great. Well, thank you for coming on.
Thanks so much for having me, Brenda and Jason, and great talking to you.
It was a great conversation. Thanks for joining.
Thank you.
This brings us to the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get the show notes, including an episode summary and links to all of the interview and roundup papers. You can also reach out to us on Twitter at @immunopodcast or via email info@immunologypodcast.com with feedback or to suggest guests. See you next time.